Thank you for coming to my digital presentation of Pore-formance of NMR cornstover measurements, where we concluded that NMR measures pore water absorption in heterogeneous media. So you might be thinking, why does this matter? Well, fossil feedstocks are commonly used to produce a lot of fuels and goods, but they result in lots of greenhouse gas emissions. Food crops like corn provide a renewable alternative, but using food for fuels and goods increases food costs. Another alternative is to break down agricultural wastes into sugar building blocks for use as precursors for fuels and goods. But the problem with this is these are really complex structures that are hard to break down. An example of the structures is shown here in this cartoon. So you can see in plant cells, there's the center vacuole or the lumen where water can be found. And then surrounding that, there's a cell wall. If we zoom into the cell wall, we can see that it's this big, complex, heterogeneous mass. And inside, there's these circular shapes known as microfibrils. There's hemicellulose surrounding that, lignin in there as well, and then some water. And then if we zoom in on a microfibril, we can see that in between individual cellulose fibers, there's also water. So you can see that at each length scale, there's characteristic water for each. And we can relate these water populations to T2 relaxation times as a way to quantify water absorption across the length scales. So what are these relaxation times? Well, here's an equation for T1 and T2 relaxation. And the important thing to note is that they're both frequency and correlation time dependent, or they have this omega naught and tau c dependence. For a fixed frequency, if we plot T1 and T2 across the correlation times, we can see that for slow molecular motion, there's a divergence of T1 and T2. For rapid molecular motion, these relaxation rates converge. Looking at a plot that correlates T1 versus T2, there's a dashed line when T1 equals T2. Things close to this line have rapid molecular dynamics, indicative of liquids or amorphous solids. Things far away from this line have slower or more restricted motion, which indicates crystalline or ordered solids. Taking the ratio between T1 and T2, or basically how close is something to that dashed line, we can see that it indicates surface mobility. So with that, let's talk about pores, and specifically water absorption within them. If we have a dry pore with a little bit of water floating around, it'll eventually hit that surface, stick to it, and kind of bounce around before desorbing. The amount of time or the length of time that it's adsorbed to the surface is known as the surface residence time, or tau1. As we continue to hydrate our pore, eventually we'll have a monolayer of water adsorbed to the surface. And so subsequent water molecules will hit and stick on top of that and start to form a second layer. The length of time that they're associated with this second layer is this second layer residence time, or tau2. Utilizing properties of these T2 relaxation times, we can actually get at these residence times, and which ultimately means that we can measure the ratio of tau1 over tau2 using relaxation. And this is useful because this can be used to estimate net heats of adsorption. So ultimately, we can estimate net heats of adsorption with NMR relaxometry. So what did we do? Well, we took corn stover, and specifically 6 anatomical fractions of it, or 6 parts of the corn plant, and we looked at 3 relative humidities. We took T1, T2 correlations at each of these hydration levels, and we can see that for each of these fractions, hydration causes a shift in the major population towards that dashed line, or it increases the molecular mobility within the system, or you could say that increases surface mobility. And so we did this for all the fractions, all 6 fractions, and we plotted that T1 over T2 ratio with moisture content. And we can see that with increasing moisture, that T1 over T2 ratio decreases, which means that adding water increases surface mobility within these materials. So that's kind of the gist of our poster, and what we wanted to get across is that the usefulness of NMR relaxometry to measure these thermodynamic properties and to estimate surface mobilities. And so with that, if you have any questions, please feel free to reach out to me at any of these links below.